analyses of biomarkers in clinical trials. Our clinical focus is on improving care for patients with leiomyosarcomas and undifferentiated pleomorphic sarcomas. All right, Elise, go ahead. Take it away for us.
Thank you. I'm going to share my screen. Yep, perfect. And yep, and I'll swap for this place. So you can see my slides, right? I'm going to take that as a yes. So thanks. So thank you to the organizers for this really nice seminar series over the weekend and for inviting us to join and share some of our thoughts on this important topic. So what I'm going to talk about are more novel therapies that have somewhat less proof and that we give in less standard of care settings. So most of what I'm going to discuss, unlike what Dr. Faruqi and Dr. Rattan were discussing, are not treatments that we give in the localized setting, but in more advanced types of diseases. So this is the outline of my talk. I'm going to start to talk about a general principle, which is the principle that we call precision oncology, on which most of our drug development is based as of right now. So I'm going to skip that first slide because you heard a lot about the different subtypes and the heterogeneous diseases that are sarcomas. So this is the theory of precision oncology. So we start with all those patients that have sarcomas, and we can see they have slight differences, but it's hard to really tease apart the different tumors. And then after a good clinical exam, pathology review, imaging review, and what we call molecular testing, we're able to see that all those patients are different and that they actually have very different tumors. And the goal of precision oncology is to then have a precise drug for each one of those patients by grouping also some of the patients that maybe have more frequent molecular subtypes. And so for all of those patients, we have one precise drugs. And so the whole theory is that for each patient, we have one type of tumor for which we have one drug. Now, in practice, unfortunately, in sarcomas, we're actually very good through clinical exams, pathology, imaging, and molecular testing. We're very good at seeing all those differences in our patients and in our tumors. We've become very, very good at grouping them together in a similar fashion. But unfortunately, right now, the reality is that the vast majority of our patients, the best treatment we have for them remains chemotherapy-based treatment for sarcomas. And there are some rare instances that we have very good signs of response for some subgroups of patients for either immunotherapy or targeted therapy that I'm going to discuss today. Our goal through notably the clinical trials and development of novel drugs is to restrict this circle and to have more patients have their own types of treatments, as was displayed in the first slide just before that. So I'm going to try to talk about targeted therapy now. Just moving to what is a targeted therapy, we're going to start with the genomic and molecular testing. So when you look at a cell, any cell of your body, on average, there are 22,000 genes. So I couldn't represent 22,000 genes. So this is a cell with the DNA and all the different genes in your cell. In a tumor cell, on average, there are 1,000 of those genes that have mutations. So, which means that the machinery of the cell is different on 1,000 types of instances. But out of those 1,000 mutations, on average, there are only 2 to 7 of those genes of those of this machinery that is really important for this tumor cell to survive and to give it its cancer phenotype. And so those are those red ones. So if I have a red gene, an essential gene that is mutated, and I have a good drug that actually acts on this essential mutation, then the drug will cause most of the tumor cells to die. If I have a good drug, but it's against one of those mutations that is more in the background, but it is not critical and essential for the tumor cell to survive, then unfortunately, most of the tumor cells will adapt. Some of the tumor cells may die. It may slow down the progression of the disease, but most of the tumor cells will actually survive. And unfortunately, in most cases today, we're able to see those mutations, but we don't have any good drugs available for those mutations. So when we focus just on sarcoma patients, this is a diagram on the left showing you the most frequently mutated genes in sarcomas, and this is just a representation, but you do not need to know those things, those names of genes. And when we look on average in all sarcomas, we find 4 essential genes mutated in a sarcoma. On average, there are 4 genes that are really essential, critical to the cell, to the tumor cell to survive. But most of those mutations, the first one being the TP53 up here, we have no drug available to target them, unfortunately, today. Now, if we look at all sarcoma patients as a whole, we have only 40% of sarcoma patients that have any type of alteration for which we have a drug. So the vast majority of patients, 60% of patients, we don't even have a drug that targets one of those alterations. But for the 40% of patients who do have an alteration that may be targetable, most of those alterations for which we have a drug are not those critical essential genes. So those drugs that we use for those 40% of patients, most of the time will just slow down the progression of the disease, may kill some of the tumor cells, but unfortunately, we'll still have some progression of disease ultimately and resistance to those treatments. So the counterexample, the example that we use the most in sarcoma and that we're aiming to develop for all our patients is the example of NTRK, which is a specific mutation that is unfortunately found only in 1% of our patients. And we've had 2 drugs being developed for this particular gene that is mutated. And on those graphs, each one of those bar is one patient. If the bar is going downwards on this part of the graph, then the tumor size of the patient has reduced. And here for those patients here, for example, it's reduced 100%. So those patients who had metastatic disease, their tumor has been totally wiped out, at least as much as we can see on CT scans, by the drugs. And so as you can see overall, all the patients who had this NTRK gene here, and most of the patients who had the NTRK gene here with those treatments had tumor reduction in size. And here the only patients who had an increase in size of their tumor were the patients who did not have this NTRK gene. So unfortunately, this is only 1% of our patients who do have this NTRK mutation, but what we're aiming to is to identify more and more of those essential genes and have drugs to target them. So now I wanted to walk you through kind of the different questions to ask yourself when you're talking about targeted treatments and genomic testing. So why would you want to do genomic testing? Well, to identify those very rare targets that are worth targeting with specific drugs. But it is somewhat of a fishing expedition, and only a minority of patients will actually have a drug to target their tumor today. The second reason, though, is to have access to novel drugs. So through clinical trials right now, we're developing more and more drugs that targets more and more of those genomic alteration that up to now we were unable to target. So in order to participate in those clinical trials and have access to those novel drugs, it will be required to have this genomic testing done. And another reason is to actually improve knowledge for other patients, as sarcomas are rare. We have rare subtypes, in helping us characterize which subtypes of the sarcomas actually have some certain types of mutations is helpful for the drug development pipeline to know which drugs we can prioritize, which drugs are worth investigating for our sarcoma patients. Why not do the genomic testing? Well, the first reason is the cost. Most of those genomic testing are now paid for if the patients are metastatic or in the advanced setting. You will have financial aid programs, but you should definitely ask the cost to your oncologist if you're getting some genomic testing, because they can be costly. So when you should do it, essentially in the metastatic or relapse setting, because that is the setting in which those drugs have been developed and for which we might start thinking of clinical trials. It's usually more useful after the first line of therapy, because that's when we start talking about those clinical trials. But we usually recommend it to do it early on, because it does take 4 or 6 weeks for us to get the results. So if we do it at the time we need it, then we're going to have to wait. When not to do it in the localized disease setting, this is not something we usually recommend in this setting. It's usually not a tool that we use to prescribe treatment in that setting. And then the question is what type of genomic testing you should do. There's a lot of companies. Overall, they all present with the same offer. I will not even give any names or advertise one a company over the other. We have one that we do most frequently at Anderson, but it's just because we're used to seeing their reports. If I have patients who came with another type of genomic testing from another company, I never ask to redo it, and that's all the information I need. Now, what you should ask your oncologist concerning those genomic testing and those targeted therapies, if you are in the metastatic or relapsed setting and you do not have and you have not had this discussion about getting the genomic testing done, it's a question that you can ask your local oncologist. Would it be worth doing? When you actually have a mutation identified, then the questions are, is there a targeted drug for that particular molecular alteration? Is this particular drug accessible in the standard of care, or is it only through clinical trials? And is the data concerning that targeted drug showing you that it's going to be more effective than chemotherapy for that particular molecular alteration? Knowing that a lot of those drugs have a very big rationale, but if you're in a clinical trial, you may not have the enough experience to say that it's going to be more effective than chemotherapy. And maybe that gene that we identified is one of those orange ones, one of those passenger ones that is not so critical for the cell and might not be more effective than chemotherapy. And then every time a new treatment is recommended and discussed, you should ask both the toxicity and the benefit expected from this drug to your oncologist. Now I'm going to move on to the immunotherapy part of the talk. Immunotherapy has truly been a revolution in cancer care over the past 15 to 20 years now. It's a concept that's been developed in 1985. The concept is that your immune system recognizes things that are foreign or that are a danger to your body. So your immune system is probably getting rid of cancer cells for you at the beginning of the disease. And then can we activate this immune system so it better recognizes the tumor cells and is even more capable of killing the tumor cells? There's been a breakthrough in 2005 with the first molecule that was developed by Jim Allison, who got the Nobel Prize, who was awarded the Nobel Prize for this a few years ago. And this molecule, which I'm going to discuss just after, its role is to reactivate your immune cells so that they recognize the tumor cells and kill the tumor cells. Now, those therapies are part of the standard of care. They are approved across multiple cancer types. Now, why are we also excited about the immunotherapy? It's mostly due to data in patients who do not have sarcomas, but mostly in melanoma, lung cancer, kidney cancer, and other types of cancers. The responses there are seen in the magnitude of 30 or 40% of patients, but what's really exceptional here is that the patients who respond can get very long-term efficacy, meaning some patients who have widely metastatic disease in melanoma or in some kidney cancers and lung cancers were able to get very prolonged tumor control, with even some patients who have stopped immunotherapy after 2 years and have not had any progression of disease for now several years. So the molecules that have been developed with those results up to now, what they do is their name is mostly anti-PD-1 or anti-CTLA-4. So PD-1 and CTLA-4 are actually receptors expressed on your tumor cells, and what they do is tell your lymphocytes, which are your white blood cells, to not attack them. So what the anti-PD-1 and anti-CTLA-4 do is actually block that receptor so that the lymphocyte is reactivated and can, in fact, attack the tumor cell and kill it. So those immune checkpoint inhibitors and sarcomas, so those are once again those same plots showing you the reduction in tumor size versus the increase in tumor size. And what you can see is that overall in all sarcoma types, the response rate is in the magnitude of 10% or less. But we do have some specific subtypes of sarcomas that have shown very good responses to those immune checkpoint inhibitors. higher than this 10% response rate. And those are essentially undifferentiated pleomorphic sarcomas, angiosarcomas, and alveolar soft part sarcomas. Now, there are other rare subtypes of tumors in which this can be indicated, and even all sarcoma subtypes if they have very specific biomarkers, amongst which the two most important biomarkers of response to immune checkpoint inhibitors that have good proof across all cancer types are microsatellite instability and tumor mutational burden. And this is because the more your tumor is mutated, the more it looks different from your own tumor cells, and the more from your own body cells, and the more your immune system is going to be capable of recognizing this as an intruder and as a danger. Both the microsatellite instability and the tumor mutational burden will be included in all of the genomic tests that we discussed during the targeted therapy part of the talk. So the other markers that I've noted here are markers that have somewhat less proof or are less standard of care, but are all signs of looking at the fact that there are lymphocytes in the tumor, and therefore they are being blocked by some mechanism in killing the tumor cells, and therefore they can be reactivated. Now, there are other types of immune therapies, all of which are only part of clinical trials and only for some specific subtypes of sarcomas. The most promising one that we're hoping is going to get approved as standard of care in the next few years are the modified lymphocytes, specifically for synovial sarcomas and for some patients with myxoid liposarcomas. What this is in synovial sarcomas and myxoid liposarcomas, those tumors express a specific molecule, and we take the patient's lymphocytes and reactivate the lymphocytes exactly against that specific molecule found on synovial sarcomas and myxoid liposarcomas, so that the lymphocytes, when we give them back to the patient, go straight to the tumor where they recognize this specific molecule and kill the tumor cells. Those therapies have been very promising over the past few years, and we think it's going to get approved hopefully in the next few months to years. Other therapies that are being developed based on immunotherapy, it's actually a combination of the immune checkpoint inhibitors with chemotherapies, targeted therapies, and with radiation therapy. The other types of therapies like vaccine cytokines and bispecific T-cell engagers and a lot of other therapies have some promise, but somehow have less data right now in sarcomas. So immunotherapy in sarcoma, why would you want to get it? Because of this long-term disease control in some cases, and somehow it does have a lower toxicity rate than standard chemotherapies. However, there are some rare autoimmune toxicities that can be very morbid, although they are more rare than toxicities in the chemotherapy setting. And also, if you have histologies that are not responsive, then you don't want to get immunotherapy because you just get the risk of toxicity without any potential for benefit. So some histologies are known to not respond at all to immunotherapy. So when you should do immunotherapy, for now, the standard of care is going to be in the metastatic relapsed advanced setting, mostly after failure of the first chemotherapy line, so in the second or third line. There are some exceptions to this rule, and angiosarcoma patients can get immunotherapy in the earlier setting in the first line, and we often do it. And in terms of clinical trials, we now have more and more data showing us that the earlier you give the immunotherapy, the more likely it is to be effective. But this is only in the clinical trial setting. However, if you have a histology that is very sensitive to immunotherapy, and there's a good clinical trial in the first line with combinations with other modalities, that may be a very good option. So one of the reasons when you should not do immunotherapy, first of all, if you have an uncontrolled autoimmune disease, so as I was saying, there are rare autoimmune toxicities. The goal of the immunotherapy is to reactivate your immune system against the tumor cells. But in doing this, it's going to reactivate most of your immune cells. And if there is an underlying autoimmune disease, it can get reactivated. And then one of the things is that immunotherapy will be active for a longer time, but takes longer to actually become active. So if there is a disease that is rapidly progressing, you may not have time to see the efficacy from the immunotherapy before the progression. So now the caveat to that is now we're having combinations with chemotherapies, targeted therapies, and radiation. And in these combinations, then maybe combo with immunotherapy makes sense. But if the disease is rapidly progressing, immunotherapy alone is unlikely to be helpful. Then what type of immunotherapy in the standard of care setting? Right now, we only have the immune checkpoint blockade treatment, but you should discuss with your oncologist, and it depends on the sarcoma types and on the biomarkers of response. So as I was saying, the immunotherapy should be given right now only in the metastatic or relapsing setting. You should ask your oncologist if your sarcoma type is sensitive to immunotherapy, as although exciting, there are a lot of subtypes that are not sensitive. So when we're recommending immunotherapy, you should ask what type of immunotherapy it is, and if it is a combination with other types of treatment. As I was saying, if you have a rapidly progressive disease, the combination with chemotherapy might be helpful. If this is a clinical trial or if this is standard of care. And as we said, always ask both about the toxicity and the benefit expected from the immunotherapy to your oncologist. Now, what I'm going to focus now is on the clinical trials, and not so much on the clinical trials that are open in terms of immunotherapy and targeted therapies, because it would be impossible to sum up everything within the 20 minutes that I'm left with, but more of the clinical trial pipeline. How are those clinical trials organized? So when we're thinking of developing a new drug in oncology, first of all, we're going to test it in test tubes and on animal models in what we call the preclinical setting, and just show that the drug actually kills tumor cells. So it's an interesting drug. Then we're going to do what we call a phase 1, and what we're going to show is that the drug is not too toxic for patients. So we're going to start with small doses, increase the doses until we reach what we call the maximum tolerated dose. But the goal of that part of the drug development pipeline is to show that the drug is safe. Then we'll do what we call a phase 2. So once we have selected the dose that we want to give, we're going to give that dose to a larger number of patients and show that, yes, the drug actually kills tumor cells. Then we'll do what we call the phase 3, which is showing that the drug works, and it actually works better than what we would normally do. Following that, we would get FDA approval, hopefully. And then there's a very important part of the drug development pipeline, which is the phase 4, which is the monitoring of the toxicities as we start to give the drug to a bigger portion of the population. So this is something that we constantly do, which is reporting of the major side effects to the FDA. So in terms of the goal of each of those phases, in the preclinical phase, your goal is just to provide rationale so that we actually move forward the drug in the phase 1. In phase 1 clinical trial, what we want is to find the maximum tolerated dose. In the phase 2, we want to show that there's some response rate. In the phase 3, that this response and the survival is actually better than what we usually do. In terms of the population that's included in the preclinical models, we're doing animal models, in vitro testing, and in the phase 1 part, we're testing this in patients for the first time. So we're doing it into a limited group of patients. It's going to be 10 to 20 patients. In the phase 2 part, it's a bigger group of patients, 20 to 200 patients, and phase 3 are usually more than 200 patients. The target population also changes. In the phase 1, we usually include most cancer types. We don't select a lot the patients, and we're just testing, once again, the safety. In the phase 2, we try to look at what patients were included in the phase 1 trial, and we're saying, well, those types of cancers seem to have a better response, so let's do a phase 2 in those types of cancers. And in the phase 3, we usually select part of the population that was included in the phase 2 in order to improve the population selection and efficacy of the drug. So this is one example with the trabectedin in sarcomas. So initially there were 5 phase 1 trials in all solid tumor types. that identified that patients with sarcomas somewhat had better responses to the trabectedin than other tumor types. And following that, there were 3 phase 2 trials in soft tissue sarcomas only. And as you can see, phase 1s and phase 2 trials are really reserved to the advanced pre-treated refractory setting after failure of all your standard of care, because at that point, we don't have a lot of experience with the drugs. So that's why we need to be sure that the patient has exhausted the standard of care drugs that for which we definitely have some signal of efficacy. And then the phase 3 trial for the trabectedin was done only in leiomyosarcoma and liposarcoma patients, because during the phase 2 trials in all soft tissue sarcomas, we identified that the liposarcoma and leiomyosarcoma patients probably had the greatest benefit from this drug. This was randomized to dacarbazine, which is another standard of care chemotherapy in sarcoma patients. So this is the historical model of the drug development pipeline, but I do want you to be aware that now early phase trials are changing, and we very frequently have a combined phase 1-2 trial. And this is to make the drug development more efficient and quicker, because every time you do a new trial, you have to renegotiate things, rewrite the protocol, go through all the administrative challenges, and it takes time. So if you combine all of this in one trial, then you're going to be quicker. So in the phase 1-2 trials, you're going to have your first patients, once again, who start with a very low dose. That is not an active dose, because we're just trying to test the toxicity. You increase the dose until you find an optimal drug dose, and then straight up expand to the cohorts that had the most benefit from the first phase of your trial. And what we often do in order to better identify those patients is we do biopsies and biomarkers as part of those clinical trials in order to identify really the best population of patients who are really benefiting from the drugs. So an example of this is actually the NTRK example that we were talking about earlier with the targeted therapies. In those 2 trials that were just phase 1-2 trials, they led straight to FDA approval, because as you can see, the response rates were amazing, with most, if not all, patients having some form of tumor reduction if they had the NTRK mutation. In this very rare subtype of patients, we can often, if we get very good response rate with a very good rationale, not do a phase 3 trial and get FDA approval for multiple reasons. If we were doing a phase 2 trial in this rare subtype of cancer, then it would take 10 years before we're actually able to give that drug to patients. And we just know that with such great response rates, this drug is going to be beneficial for this population of patients. And also, when you have such great results, it's not ethical anymore to a certain extent to randomize patients in a phase 3 to something that we know for sure is not going to be as good. When we do phase 3, the understanding is that we don't know for sure that the drug we're testing is better than what we were doing before. So now that we're focusing a bit on early phase trials in oncology, as we said, there's less data, but there has been a lot of improvement in early phase trials over the past few years. And I want to bring your attention on this graph. Here you have the response rate, and here is the year of the development of the early phase clinical trials. So whereas in the year 2000, we had a little less than 10% of patients included in the early phase clinical trials that did have some form of response rate. Today, we're more in the 30 to 40% response rate for patients included in early phase trials. And that is because we have become more efficient at identifying good biomarkers and targeting those essential genes that the tumor cells rely upon. We do have to be honest that there is still a 1%, around 0.5 to 1% toxic death rate in those early phase trials. It has remained stable over the past few years with a tendency to be lower, but it is there because, once again, we're testing new drugs, and so we're unclear on the toxicity in humans. Now, as I was saying, the genomic biomarkers have increased our response rate to early phase clinical trials. So while the response rate to all early phase clinical trials is around 20 to 30%, if you look at those clinical trials that have a strong biomarker and that are selected for this biomarker, the response rate is more in the magnitude of 30 to 40%. Now, moving on to specifically sarcomas, then we do have data, 2 papers showing us that if you're going to participate in an early phase clinical trial, then the clinical trials that are based on a molecular target or specifically on your histology type of tumor, then the benefit of the clinical trial is going to be way bigger. So to sum up the differences between what we call the early phase clinical trials and the phase 3 clinical trials, on the one hand, in the early phase clinical trial, all the patients will actually get an active drug. However, if you're at the beginning of the phase 1 trial, you might get an ineffective dose because we're just testing the toxicity. Now, if you're getting towards the end of the phase 1-2 trial, then most patients actually get an effective dose. Most trials, although not all right now, will authorize patients who got the ineffective dose to start with to actually increase the dose as the trial is accruing. But you do want to ask this to your oncologist before participating in an early phase trial. Now, the benefits is that those drugs are more novel and usually more biomarker driven. And the biggest risk is that we do have less data. We have less patients that have had the drug. The benefits of a phase 3 trial is that we do have more data, that you're sure that you're going to have an active dose. The risk is that you're going to be randomized, and you're going to be randomized to the standard of care, which might be less effective, but we don't have enough data to say that it's less effective. Some randomized trials are actually randomizing to placebo, and this is really something you want to discuss with your oncologist before participating in a clinical trial. So clinical trials in sarcoma, why would you want to participate? Well, it gives you access to more novel drugs. After the second line of chemotherapy in sarcomas, as a general rule, the response rate of early phase clinical trials and phase 3 clinical trials is likely to be superior than most of our third line treatment options. And I say most because there are some histologies that are very responsive to standard of care drugs, and you do need to discuss this with your oncologist. But after 1 or 2 lines of chemotherapy, clinical trials are a very valid option. And then, of course, when you're participating in clinical trials, you're also improving our knowledge for other patients. Why not to participate in a clinical trial? The biggest reason is it's going to be very, very time consuming. You're going to have to come more often in the hospital. There might be biopsies. There's going to be more lab work. So you do want to ask your clinical trial, your research team before participating in the clinical trial, exactly what is going to be the schedule of the visits, the biopsies, and the scans. Of course, in clinical trials, we are not fully aware of the toxicities. So when you should participate, it really varies, and this is something that needs to be discussed with your oncologist, and it depends on your tumor type, on the available clinical trial, the data on it, and what is the data on your standard of care drugs. So when you should not participate, if you really have a very good standard of care option, usually with a response rate that is expected to be more than 20%, then unless there is a very, very good molecular rationale, probably the standard of care option is good. Of course, if there's no trial available, but today we have so many clinical trials open, some molecular targeted, some that are not molecular targeted if we don't have a target for the genomic alterations identified. So I think having no trial available is a rare finding. Mostly if you're too tired, then no trial will be available. If you are too tired from the toxicities of prior treatment, and then the risk of toxicities of those clinical trials is big. Now, what type of trial? If you have several trials that are presented to you, you should mostly go for those that are histology selected, specific to your sarcoma type, or that are biomarker selected for the specific molecular alterations that we have identified. And then if you're going for an early phase clinical trial, then you want a clinical trial that you are going to receive an active drug. Now, what you should ask when your clinical oncologist is talking about clinical trials, first of all, at any time you can initiate the conversation on the possibility of participating in a clinical trial. What are the clinical trials that are available? And I do think that in the metastatic and relapsed setting, you should ask for biomarker screening through genomic testing as early as possible, because even if we don't identify at that time a biomarker for which there is a clinical trial, this is a moving landscape and there might be a clinical trial for that target in 6 months from now. And just having the genomic testing already done will be helpful for inclusion in those clinical trials. So when you are recommended to participate in a clinical trial, do ask what phase of the clinical trial it is. Ask if it's a histology-specific or a biomarker-based clinical trial, as those usually have higher response rate. Do ask if there is a standard of care option, and this is something that should usually be presented to you in the same time that you're presented with a clinical trial, is what are the other options, and is there another option? And then ask about the schedule. Once again, those trials are very time consuming. Ask if you will be given an active dose and if this is randomized, because those are things that patients may feel comfortable with or not. Some patients are not comfortable with being randomized in a trial, and that's perfectly reasonable. So my conclusions are that there are a lot of novel drugs being developed, and that we're hopeful that we'll get to that goal of precision oncology, of having novel drugs for all those targets that we're identifying. But for the moment, in the vast majority of cases, chemotherapy remains the standard of care for sarcoma patients in the first-line setting. Genomic testing should be considered in the metastatic and relapsing setting, as it can give you access to targeted drugs, also to immunotherapy, to clinical trials, and this would be given after the first line of chemotherapy. Targeted drugs are currently available for a vast minority of patients, but there are a lot of novel drugs being developed in that space. Immunotherapy is active in select sarcoma types. There are new combinations being developed, and clinical trials and targeted clinical trials will give you access to those targeted therapies and those new immunotherapies. But you do want to discuss the choice of the clinical trial and the molecular profile with your oncologist. Thank you. And I will take questions. I think I'm on time, actually.
Perfectly on time, actually 2 minutes ahead. Nice job. Thank you so much, Dr. Nassif. I know there are quite a few questions in the chat box here. Are you able to access those, or would you like me to read them out?
I'm gonna go. Oh yeah. Okay.
What
does genomic testing entail? I'm in the UK. I'm not sure where to go to get genomic tests. Oh, Dr. Rattan answered that one. Dr. Rattan answered a lot of those. Okay. Can radiation, this was already answered. Sorry. I don't know where I am. Dr. Rattan answered a lot of those, I guess. So, what are the results you are seeing with MDM2 and CDK4/6 inhibitor for well-differentiated retroperitoneal liposarcomas? All MDM2 inhibitors are not equal. All CDK4/6 inhibitors are not equal. Some are probably more effective than others. The only one that is standard of care option right now is the palbociclib. The MDM2 inhibitors and CDK4/6 inhibitors as part of clinical trials are both very promising. I would say that the MDM2 inhibitors are somewhat more promising for the well-differentiated retroperitoneal liposarcoma patients right now. But CDK4/6 inhibitors, there's a trial, multi-institutional trial with abemaciclib right now. And I think for liposarcomas, this is a very good clinical trial option. Genetically tested positive, so STAT6, yeah, this is the part of the SFT diagnosis. Would you expect to see the same results? Sorry, I'm missing that. It's moving. On subsequent testing, or can it change over time in different tumors and with different treatments? So this is a very good question. If you're getting targeted drugs for your particular alteration, then yes, it can move, it can change. But overall, with chemos, we see that it's very stable over time. If you've had several lines of therapy between your genomic testing and your inclusion in a clinical trial, then it's a valid thing to ask to get retested. But other than specifically targeted therapies, you should not get a mutation that would make you resistant to the targeted therapies if you've been treated with chemotherapy. But that is a very good question. And as part of the clinical trials, more and more frequently, we are doing repeated biopsies to look at this very question. Okay, I think this has been answered. So there are multiple good cancer centers over Europe. In Netherlands, there are a few. I'm gonna miss the names. In Europe, there are a few. I can't give you all the names. In Europe, you have some in France and in Italy, in Spain. In Netherlands, you have very good cancer centers, and in England. You should just be attentive to the fact that there are sarcoma centers. So I know the names of the doctors, but not their centers, unfortunately. I'm sorry. Let me see if I can find you the name. I'm going to forget the name in the Netherlands. It's the Netherlands Cancer Institute, so it's very easy. And in Europe, you have Gustave Roussy in Paris, you have Royal Marsden in London. There's a in Milan, they have a very good cancer center with a sarcoma specialist. I don't know that I. So, as within the Department of Sarcoma Medical Oncology at Anderson, I don't think that we do have clinical trials for PICOMA patients as of right now. But we do have a lot of clinical trials open in the Investigational Cancer Therapeutics Department, and there would surely be a clinical trial, potentially not specifically for PICOMA patient, but that would qualify. Okay.
All right. Any final questions? Please go ahead and add those to the chat box. We have a few more minutes here.
Do you see future clinical trials with new immunotherapies opening to patients who have already tried different immunotherapies in the past? Yes. Yes, absolutely. Most immunotherapy trial combinations right now, so not in the sarcoma space, but that you would qualify for in the early phase trials that they're opening to multiple patients. We have most of those clinical trials are open to patients who already have immunotherapy. The only caveat to that is if you have had a big toxicity on previous immunotherapies, then you would not qualify for any new immunotherapy trial. But we have multiple immunotherapy combinations that are open to patients with several lines of immunotherapy prior to that.
All right. I think that's the last question. Oh, there's one more.
In the charts, I saw responses and non-responses for liposarcoma. What is the general perspective? Is immunotherapy a viable option for advanced liposarcoma? Very good question. So I would say that initial trials of immunotherapy have identified liposarcoma as being somewhat more responsive, but the more patients we've included in these trials, the lesser the response was. So there is probably a subgroup of patients with liposarcomas, that do benefit from immunotherapy, and we don't really know yet how to select those patients. And maybe by combining with other therapies will increase the efficacy of the current immunotherapies. but it's a very good question and one on which we don't have a full response right. So for clinical trials, some clinical trials are opened nationally across several institutions. So if you come to Anderson, but the trial is open to a closer center to your home, I would definitely let you go closer to your home where this center is open. Some clinical trials will not be open in the center close to you. And because at least for the first month or two on a clinical trial, you're going to have to come almost on a weekly basis. We prefer you to have to stay around for a month or two. And then once the visits spread out on the clinical trial, you can go back and forth, but you actually have to continue to come to the center in which you're enrolled. So that's definitely one of the challenges of clinical trials, but some clinical trials, most clinical trials are opened at different sites. And so we can try to find a site in which it's open closer to your home. Yes, there is a trial using cabozantinib with immunotherapy drugs. It's a very promising combination. The toxicity rate is a bit higher, much higher than immunotherapy alone, but I think it's a very promising combination. One of those trials is actually going to be open, is just got activated through the NCI, so will be open in multiple cancer centers across the United States. It is going to randomize patient between cabozantinib and a combination of two immunotherapy versus the combination of two immunotherapy drugs. And we're going to open it at Anderson in the next few months.
All right. Any